What digests starch? Thank you, Abhishek. Thank you, Abby. Amylase. You can say protease, not protease, sorry. You can say carbohydrates as well if you want. But the one specifically for starch is amylase. So this is to do with the digestive system. So it's a type of carbohydrates. There we go. Amylase, a type of carbohydrates that breaks down starch. Where are most food molecules absorbed? Digestive system. This is the digestive system. So mouth, esophagus, goes down to the stomach, then through past the liver and the pancreas into the small intestine, which is the answer to the next one. Well done, guys. Small intestine. And then we've got 2 villi here. Figure 2 also shows one cell of the surface of a villi. So villi is what you find within the small intestine. So that comes under here, diffusion. I've got it here somewhere. Yeah, there we go. So inside the small intestine, can you see that there? It's not smooth. It's not like a smooth tube. Within that, there's these little structures called villi. And we've got one advantage of using an electron microscope compared to light microscope. One advantage. Anyone know? Bigger magnification. Say bigger resolving power as well. We'll come back to the villi. We've got the question on the villi in a second. What type of blood vessel is labeled X? We've got 3 types of blood vessels, guys. Arteries, capillaries, veins. Which one would you think X is then? Arteries carry blood away from the heart. Vein carry blood into the heart. Capillaries basically join arteries and veins together. Yep, DP, well done. Capillaries. So the arteries basically break up into capillaries. So this here might be an artery. So it breaks up into the capillaries. That's the artery. Let me get rid of this bit. Breaks up into a capillary. And then, can you see it comes back into this one, which is a vein. And then it carries on through the vein. So it goes artery, splits up into capillaries, then it comes back together to form the veins. Next question. Next question is one about magnification. So we've got the real length as 0.8 millimeters, magnification of times 20. They've literally given you the equation. So you can just put the equation, the numbers in. Do we need to rearrange this? Yeah, we need to work out size of the image. Size of the image is equal to magnification times actual size. So let's multiply the 2 numbers together. 20 times 0.8. That gives you an answer of 16 millimeters. Figure 2 shows 2 cells from the surface of a villus. There are sugar molecules inside and next to each cell. Name the process by which sugar moves into cell A, and then it asks into cell B. What's the difference here? Cell A and cell B. So this trying to get into here, and this trying to get into here. Which one's going to be easier? What are the 2 processes called? So here you can see there's loads of sugar particles. So we've got a high concentration. Here we've got a low concentration. Whereas in cell B, we've got a low concentration outside and a high concentration inside. So what are we talking about here? So cell A is high to low. Cell B is low to high. No? No one remember? Yep, well done, Harry. It's diffusion for the first one. There we go. And second one is active transport. Okay, well done, guys. It's not osmosis because osmosis is to do with water. Okay. Osmosis is like diffusion, but to do with water. Because this is to do with sugar, the answer is not osmosis. Give one use of sugar in the body. Sugar is basically glucose. What could you say? One use of glucose. We use it for energy. So yeah, for respiration. Good. Respiration. You can say other stuff as well. Glucose is stored as well, isn't it? It's stored as glycogen. So you can say that as well. It's stored as glycogen in the liver. You can also say that you use the glucose to make amino acids. You can say that as well. But yeah, the main thing you should think is respiration whenever you think about sugar. Right, so we got a 4-mark question here on the villi. Explain how the villi are adapted for efficient absorption of sugar molecules. So you got to use your own knowledge and also try and use what they've given you in the question. What do you guys think you can say here? So they've zoomed into one cell. They've labeled mitochondria, so it's probably good to mention this. And they say that each villi have these microvilli sticking out of it. What do you guys think you can say here for these diagrams? 4 marks. Let's try and write 4 points here. So we should probably mention the stuff that they're talking about here. So let's talk about mitochondria. Why would we need mitochondria? And what can we say about the microvilli? And then what else can we say? Try and think about that. Anyone know? Here, I've got the notes here. So we have the fact that it's a single layer of surface cells and blood capillaries. So it's got blood, it's got a good blood supply. You could mention that as well. You can tell that by X. Remember, these were the capillaries. What do you guys think the microvilli do? Why is that good? The microvilli. Larger surface area. Well done, Harry. So it gives it a larger surface area. Mitochondria, why would it be useful for these to have mitochondria? These are the cells. Remember, villi are found in the small intestine. And they are used to absorb digested food molecules. So if you have this situation here, where the digested food molecules are low concentration, and that you're trying to get it to go into a high concentration, you're going to need energy, aren't you? Where do you get that energy from? You get it from the mitochondria. So that's what you should mention there. Why do we have mitochondria? To transfer energy for respiration, so that energy can help the active transport for respiration. So you can say for respiration to transfer energy for active transport. I think we've covered 2 points there. Microvilli, large surface area, good blood supply. What's good about that is that it increases the diffusion gradient, maintains the diffusion gradient. We've got more than enough points here. Concentration gradient. Yep, Mary, well done. You can mention thin lining, or you can say the walls are 1 cell thick if you want a fourth point. And what that does is it basically decreases the diffusion pathway, the distance that diffusion needs to occur. So it decreases the diffusion pathway. Okay, yeah, we've got more than enough for full marks here. You don't need to mention all of that. I just thought I'd go through everything so that you guys know. Okay, well done, guys. So on to the next one. Water is lost from the leaves of plants through the pores called stomata. What is the loss of water from a leaf called? Just because it says water here, it doesn't mean the answer is osmosis. It's not osmosis. Osmosis happens through a partially permeable membrane. There is no partially permeable membrane. The stomata are holes that you have on leaves. So yeah, well done, Abby. Answer is going to be transpiration. Well done, Ashni. Not respiration either. Which cells control the size of the stomata? Who knows that one? That should be an easy one. Let me get this topic up. Plant tissues. Yep, well done, Abby. Guard cells. So you got guard cells around the stomata, which open and close. See that? If you zoom into a leaf, you get all of your stomata. And then surrounding the stomata are the guard cells, which can open and close. That's that one. Right, on to the next one. A student investigated water loss when different surfaces of leaves were covered in grease. The grease blocks the stomata. So these holes that you have, when you add grease to it, it closes those holes, okay? So what they've done is they've taken 4 leaves from one plant, they've put grease on different surfaces. So you can see here, they've put one on the upper and lower surface. This one, they've only put it on the upper surface. Here, they've only put it on the lower surface. And here, there's no grease at all. That's the control of our experiment. We're using that to compare. And what they've done is they recorded the mass at the beginning. Then they let the leaf just dry for 24 hours. And then they recorded the mass again. So what must have happened in 24 hours is transpiration. So the water must have evaporated from the surface of the leaf. And the mass of the leaf would have decreased. So that's what you can see in this table here. Loss of mass. That's because the water is leaving. So all of them have decreased. So first one, calculate a value for X in the table above. This is the mass that it starts off with. This is the mass that it ends up with. So all you got to do is minus the two numbers. 2.01 minus 1.97, which gives you an answer of 0.04 grams. The loss in mass of water was measured after 24 hours. Calculate the mass of water in grams per hour. What do you guys think you need to do here? 2 marks here. So after 24 hours, we've got our loss of mass here. For leaf D, we want to work out the mass of water lost per hour. So leaf D is this one here. Any idea what we need to do? So 0.15 grams lost in 24 hours. We want it per hour. So that basically means in 1 hour. Yep, well done, Hanya. You get 0.15 divided by 24. And that's going to give you per hour. So when you work that out, you get something like this. You get 0.00625 grams. On to the next one. The student concluded more water is lost from the lower surface of a leaf than from the upper surface. What evidence is there in the table to support that conclusion? More water is lost from the lower surface than the upper surface. Is that true? Yes, it is, because look at this one. Whenever the lower one has been covered, remember these, if it's covered in grease, it means the stomata are no longer open. So here, when the lower one is covered, it's the mass that's lost is very low. Here, the upper and lower are covered, and we calculated that as 0.04. Let's write that in here. So this and this, so leaf A and leaf C are the ones where the lower one was covered. So it does support that, because whenever the lower one was covered, whenever the lower surface of the leaf was covered, less mass was lost. So we can say that whenever the lower surface of the leaf was covered, less mass was lost. So what did the results in the table above show about the number of stomata on the surfaces of the leaf? So whenever the lower surface was covered, less water was lost. So that means the lower surface must have more stomata. And it's true. You have much more stomata at the bottom. The reason why is because the plant doesn't want to lose too much water. It's bad for it if it loses too much water. So if it was at the top, then it'll get a lot of sunlight. It'll evaporate much more quickly. So there's more stomata at the bottom because that's in the shaded part of the leaf. So less water gets lost. And that this proves it, because whenever the bottom one was covered, less water was lost. So that must mean there's more stomata at the bottom. Hope that makes sense. Let me know if it doesn't. I'll explain that again. The investigation was done at 20 degrees. How would the mass of water lost be different in the investigation was done at 25 degrees? So they've increased the temperature. What would you expect to happen? So remember, this is all to do with evaporation, right? The water inside the leaves are evaporating. So if the temperature was higher, there would be more evaporation. Therefore, there would be more transpiration occurring, more water lost. So the difference in this investigation, all these numbers on the last column, they would be much greater. So you might get like, I don't know, 0.6 here. You'll get even more here. You might get 1.2, you get 0.5 here, 1.3, I don't know, something like that. You're going to get much more water loss when the temperature is higher. So you can say more water or more mass, isn't it? That's what we're measuring. More mass will be lost. Reason for that is because there'll be more evaporation. There'll be more evaporation, or you can say more transpiration as well. Okay, next one. Cells are basic units of all forms of life. 4 differences between a bacteria cell and a plant cell. Okay, let me know in the comments, guys. Let me get that up in the notes. Difference between a bacteria cell, which is a prokaryotic cell, and a eukaryotic cell. What do you guys think the answer would be? This is the first topic in biology. Everyone should know this well. Key differences between prokaryotic cells and eukaryotic cells. Still don't have any answers. Surely you guys know. So here's a prokaryotic cell. Look at all the labels. Yep, well done, Ashni. Plant cell here. Eukaryotic. So one thing, it doesn't have nucleus. Plants, actually, bacteria cell does have a cell wall. Look here, it does have a cell wall. They both have cell walls, so you can't say that. But you can say bacteria cell has no nucleus. What else can we say? So saying that the DNA is free flowing is basically the same mark. Why have I spelled it like this? It's basically the same mark as saying it's got no nucleus. Look at the other things you can say. Plant cells has plant cell has chloroplasts. Plant cell has vacuole. Yep, TP, you can say that as well. Or you can say bacteria cell has no vacuole. That's fine as well. One more thing you can say, look here, we've got these little rings here called plasmids. Bacteria cell has plasmid. Good, good. On to the next one. Gonorrhea is a bacterial disease. A new vaccine is being developed against gonorrhea. Describe how a vaccine works. 4 marks. How a vaccine works. Give me some points. What do you guys think? It comes under diseases. So you inject, remember this, dead or inactive pathogens. Yeah, well done. Yeah, that gives you 1 mark. And then you don't need to mention the antigens, but you should mention antibodies are produced. Abhishek, when it comes to contact the immune system remembers. Don't say it remembers. You get a mark for saying it produces antibodies quickly, but you don't say it remembers. Don't say it remembers. You got to mention memory cells though. So let me explain what happens. When you put the dead or inactive pathogen, you've basically you're making the body think that there are that there's a real disease in there because of the antigens. The antigens are the things that surround the pathogens. So the body will release, or the white blood cells will release antibodies to fight that pathogen. Okay. So once it's fought off the pathogen or the dead or inactive pathogen, some of those white blood cells remain in the blood. And those ones that stay in the blood here, they're known as memory cells. Don't say they remember. Those are the ones that stay behind. So just in case the real pathogen was to enter, it can rapidly produce the antibodies. So it can rapidly produce a large amount of antibodies. That's how it works. Everyone's liking Abhishek's answer. The memory lymphocytes retain the information from the antigen. Yeah, they do. They do. So what you can say, white blood cells. So you can mention white blood cells, or you can say lymphocytes. Produce antibodies. And then the white blood cells, I'm just going to write WBC, remain as memory cells. So if the pathogen was to re-enter, antibodies will rapidly be produced. Okay, so we get a mark for this, we get a mark for this. Memory cells you get a mark for, and if antibodies, and they say it will rapidly be produced. Okay, next. Let me know if there's any other questions. Another disease caused by bacteria is salmonella food poisoning. In the UK, chickens are vaccinated against salmonella bacteria to reduce the number of cases of food poisoning. Explain how vaccinating the chickens reduces the number of cases in salmonella. How many questions? Oh God. Okay, I've got to move on. How will vaccinating the chickens help? It just means that there'll be fewer bacteria. Yeah, because you're basically killing any bacteria that gets into a chicken. So if there's fewer bacteria in the chickens, it means there's going to be fewer bacteria eaten by humans. Give one way that the spread of salmonella food poisoning from one human to another is controlled. Do not refer to vaccination. What can you do with salmonella? It's basically the one that spreads when you're cooking, isn't it? It's very simple. You can just say wash your hands before you prepare food. The number of cases of salmonella food poisoning is usually higher in summer than winter. Okay, why do you guys think that is? Why is it higher in summer? Why is it higher chance you get food poisoning, salmonella food poisoning in summer? Remember, salmonella is caused by a bacteria. What do you guys think you can say here? 1 mark. Anyone? In summer, why are you more likely to spread? Temperature is high enough for bacteria to survive. They can survive in cold temperatures as well. Yeah, Mahania. Because the bacteria grows faster in heat. They reproduce faster. That's why. Tina, wash hands is for the previous question. Maybe your video is lagging. But yeah. Well done. This question is about photosynthesis and food production. How can oxygen production be used to show the rate of photosynthesis? Important word there, rate. So remember, photosynthesis, equation for photosynthesis. Here we go. Produces oxygen, yeah? So the more oxygen it produces, actually it's not the more oxygen, it's the faster the oxygen is produced, the faster the rate of photosynthesis. So what you do is you basically look at how fast it's producing. You're not looking at how much oxygen is producing. That's important. Don't say more oxygen means more photosynthesis. It's the faster or the more oxygen is produced in a given time, the faster the rate. More oxygen produced in a given time means a faster rate. Yeah. I think it is lagging because all your answers are like 5 minutes after the question. So maybe just leave and come back and then you'll be in time again. Scientists investigated factors affecting the rate of photosynthesis in tomato plants. They were growing in a commercial greenhouse in the UK during winter. Okay, so we've got 3 graphs here. This is all to do with the rate of photosynthesis and limiting factors. Name the factor that is limiting the rate of photosynthesis at point X. What do you guys think? I bet you there'll be loads of wrong answers here. There's only 3 factors that affect photosynthesis. Light intensity, carbon dioxide, temperature. There's 3 that you need to know anyway. What do you guys think would happen? What would be the limiting rate? Sorry, what would be limiting the rate of photosynthesis at point X? Any answers? No? CO2 concentration. So loads of people would put CO2 concentration or temperature, but it is not that. The answer is actually light intensity. It's a trick question. Because they said at point X, okay? Limiting factor, so this is a graph of light intensity against rate, right? Temperature and CO2 concentration are the limiting factor when it goes flat. this part of the graph. This is asking about point X when it's increasing. What does limiting factor mean? Limiting factor is something that stops photosynthesis from happening faster. What we're doing is we're increasing light intensity to make it go faster, isn't it? So in that experiment, we're keeping everything else constant. We're not changing these. So light intensity would be the factor that's limiting the rate of photosynthesis because it's not asking about the flat bit. If it was talking about the flat bit, then you can talk about the CO2 and temperature. Farmers growing tomatoes commercially try to control the rate of photosynthesis and make maximum profit. A farmer can control the temperature and CO2 concentration in a greenhouse. Okay. What is the minimum light intensity a farmer should use to get the maximum rate of photosynthesis? So for that one, you want to see the highest point on the graph, right? You want the highest photosynthesis, rate of photosynthesis. And we said that the more oxygen that's produced, the faster the photosynthesis. So that's basically this point here. That's the fastest one because the question specifically said the minimum light intensity. So anything greater than 20,000 is going to give you a maximum because that's all up here, right? Nothing goes higher than that point. Minimum would be 20,000. So the answer for this one is 20,000. 20,000 lux. The light intensity you gave in part C may not give the farmer maximum profit though. Explain why. Keyword here, profit. Why would it not give maximum profit? It will give maximum rate of photosynthesis, not maximum profit, because you've got to think about money here. It's going to cost money to run the greenhouse, right? So having a higher light intensity, having higher CO2, having a high temperature, all of that's going to cost money. That's going to basically eat into his profits. So for 3 marks here, you can mention some of these things. You can say heating the greenhouse costs money, or you can say it's expensive. Was it the light as well? Or we'll say using lights. Is expensive or costs money. Therefore, it could exceed the profit. So the costs could exceed the profits. So that means there'll be less money. So even though there's maximum photosynthesis, that doesn't mean the farmer's going to get maximum profit. Explain the result when light intensity was 0 lux. Okay, have a think about this. Look at 0 lux. Because usually you would expect this graph to be 0 at 0, but look at this. It goes below 0. Does anyone know why? You got to use your own general knowledge here. Why is it at 0 light intensity, the amount of oxygen, the rate of oxygen is negative? That's okay, Varshan. No worries. I don't care. Explain the results when it was 0. Any ideas? All right, Varshan, answer this now. I know you're bleeding. Why is it negative? No? Yeah, they take in CO2. No, nothing to do with CO2. Remember, this is all to do with oxygen. Zero light intensity. Think about how much photosynthesis is there. There's no photosynthesis. The question is, why does the rate of oxygen production become negative when light intensity is 0 in this graph? So there's no photosynthesis. So what else is there? Negative, so that means oxygen is being taken in. What process could be taking in oxygen, which causes it to be negative? No one? There we go. Well done, Abhishek. Respiration. Remember, plants also respire. So there's no photosynthesis, so no oxygen is being produced. Instead, oxygen is being taken in because the plant is respiring. So it's negative. Autocorrect, yeah? That's what you meant to say. So for how many marks? 4 marks. Oof, 4 marks. So you can say no photosynthesis. Occurs at, was it 0 lux, wasn't it? So no oxygen is produced. But instead, respiration occurs. So oxygen is taken in. Meaning it's negative. Okay. I have got 12 minutes. Okay, I better hurry up. How much do I have? Okay. Nearly done. New drugs are tested and trialed before they can be licensed to treat patients. The graph below shows how much time different stages of testing took for one drug. Okay, so they've got clinical testing and preclinical testing. How much more time did clinical trials compare to preclinical? So clinical trials and preclinical trials. That's clinical trials there. Sorry, that's preclinical trials. It's from there to there. Clinical trials is going to be from here to phase 3, end of phase 3. So that would be around here. So that's exactly 10. So we've got 3.5 here to 10. So preclinical testing is taking 3.5. That's this distance here, this length here. And then clinical testing is from 10 to 3.5. So that is 6.5. How much more time did it take? 3 hours more. So the answer is not 6.5. People would get that wrong. It's a trick question. It's not 6.5. It's how much longer will it take? How much more time will it take? The answer is 3 years, because 6.5 is 3 years longer than 3.5. During phase 1 clinical trials, low doses of the drug are tested on healthy volunteers. Your session will be ending in 10 minutes. Okay, remember guys, if I reach 10 minutes, it's just going to cut off automatically. Let me try and do everything in 10 minutes. Suggest one reason why low doses of the drug are used in phase 1 clinical trials. Low doses, basically to look for side effects. Two reasons why healthy volunteers are used. So in clinical trials, before they test it on patients, they test a drug on healthy volunteers. Why did they do that. That's because it could be like it could be a big risk. It's a bigger risk to test on an ill patient first. There's less risk testing on a healthy volunteer. What else could you say? Oh, side effects. It might be easier to identify side effects because a healthy volunteer doesn't have any other conditions. So it's easier to identify side effects. Next one. The results for clinical trials can only be published after peer review by other scientists. One reason why the results must be reviewed to avoid bias or to prevent false claims. Yeah, if someone's doing a drug trial, if a company's doing a drug trial, they're going to want their own drug to do well, isn't it? So that they can make more money. So it needs to be reviewed by someone external so that they can, so that they're not biased. Last question, 6 marker. Right, we've got 8 minutes to do this. A drug is only licensed for medical conditions it was tested on to treat in the clinical trials. Okay. Drug regulations control what drugs a doctor can prescribe, ensure doctors can prescribe a drug with confidence, and protect patients. Okay, fine. AMD is an eye condition, so that's an eye condition that can result in poor vision. Okay, 4,000 cases. Fine. And then to treat this disease, AMD, they've got drug A and drug B. Okay, so the UK used 2 drugs, drug A, drug B, to treat AMD, which is just an eye condition. They've just told you that. In many countries, there's another drug called drug C, which is also used to treat AMD. But in the UK, we use drug C to treat cancer. We don't use it to treat AMD. We use it to treat cancer, it says, but not to AMD. That's what the question's saying. And then they've given us the price. Drug A is this much, drug B is this much, and you can see drug C is much cheaper. Okay, I'm hoping you guys are understanding this question. This is something completely new. Like they can do it, they do this every year. They give you something completely new and you got to figure it out. You don't need to know anything about AMD. They've literally told you in the exam. It says in 2018, the High Court in UK gave permission for drug C to be used. So this new drug, which we use for cancer, the court has allowed us to treat this condition called AMD. Evaluate the decision to allow the use of drug C. Evaluate the decision. So is it a good thing? Is it a bad thing? If it says evaluate, you're going to have to give both sides of the arguments. So for and against, just list as many things as you can. They've given you information here. Use the information they've given you. So what's a good thing about using drug C to treat AMD? Main thing is here. Look at the price. It's cheaper. So that means whenever it comes to like medicine or medical field, think about the NHS. It's cheaper, so it saves money for the NHS. That's one thing. You can do some calculations here. How much cheaper is it? So to drug A, you can do 561 divided by 28. It's about 20 times cheaper. So you can treat 20 more people with one injection. Sorry, you can treat 20 more people. Yeah, you can treat 20 times more people with drug C. That's another good comparison you can make. Try and do some calculations if they give you numbers. 20 times more people. God, I've got 3 minutes left. With drug C compared to drug A. And you can do a similar calculation for drug B. So you can do 800 divided by 28. That gives you around 29. So you can say, and 29 times more people than drug B. Other things you can say, it's been used in other countries, so it must have already been checked, isn't it? So other countries must have tested it. Have tested its effectiveness. There's more reasons for than against. Tested effectiveness and how safe it is. Yeah. And then for against, what could you say against? It's not been tested in the UK. It's only been tested for cancer, it says. Because something's only licensed if it's been tested for that disease. It hasn't been tested in the UK. That's a bad thing. Therefore, you can't be sure it is safe, and you can't be sure it is effective. So even though other countries are using it, yeah, you can assume it's safe, but it's not completely sure until we've tested it ourselves. And also, there could be like side effects because we haven't tested it ourselves as well. Could have side effects, and all of that should be fine. And then you could just make like a decision, just be like, therefore it's a good thing, or therefore it's a bad thing. Doesn't really matter what your final decision is.